iBET is a not-for-profit research institution with a strong base of knowledge and competencies in the production, purification and characterization of biopharmaceuticals, including ATMPs and vaccines.
iBET DNA is to promote the links between academia and industry. We want to predict the needs that our partners will have in what concerns R&D. One of our most precious assets is our people. We have programs specific for training in developing countries and special programs to receive students from developing countries, and in partnerships with the Nova University of Lisbon and other academic institutes in Europe.
iBET has strongly invested in high-quality infrastructure, which included the revamping of the late-stage R&D and bioproduction unit in 2019, and the current expansion of its premises with the new iBET Biopharma building, which will provide new opportunities for iBET to innovate and succeed within its area of expertise.
iBET areas of expertise include bioprocess development for the production of ATMPs and biopharmaceuticals. Through all the process of upstream and downstream, characterization of very complex products like biopharmaceuticals, ATMPs, and vaccines, by having this full range of capabilities, we can provide biotech solutions globally in these fields of gene, cell therapy, and vaccines.
These multiple disciplines, this knowledge across a wide range of areas, and iBET's history of cooperating with our strategic partners meant that, when needed most, we were able to rise to a challenge like no other.
After having started to work with monoclonal antibodies in the late 80s, iBET went into viruses for vaccines and gene therapy through partnerships. This learned us some competencies on adenoviruses, which we then used, for example, to produce a vaccine to control rinderpest or eradicate rinderpest in Africa. Currently, we're also being part of the pest epitheliumina eradication also in Africa. They were our launching partners to produce material for phase 1 and 2 clinical trials. And one of them is currently being tested in phase 2 for cancer vaccines. More recently, we've also done a project which we labeled Divine, so we created digital chromatography processes.
Having always been based around the concept of integration in all forms, iBET has found its relationship with one particular partner especially fulfilling.
We are currently working with a European Vaccine Initiative in a project funded by the Japanese GHIT, including Heim University and Sumitomo, in a project targeting malaria. More recently, we are partners with EVI and a few others, trying to push the transvac concept one step further ahead. The idea being we need a good infrastructure in Europe that is able to take from the labs, which are more basic science driven, into vaccines that may create cures for lots of diseases already in place or the new pandemics that will suddenly arrive. It's currently an alliance, and we're very happy to be part of a campaign to support the work of the European Vaccine Initiative.
And with this ethos of shared knowledge and complementary skills, there have been many moments to feel proud of.
We at iBET have been proud to participate in very timely and challenging projects. The Serology for COVID project, which aim was to develop a serological test for detection of SARS-CoV-2 infection in the Portuguese population. We have been also participating in an European-funded project named EduFulVac. In this project, we have developed more than 30 different vaccine candidates that were later tested in preclinical animal models, such as ferrets and non-human primates. And we hope to have made a difference in the development of bioprocesses for the generation of new vaccine candidates.